Hi, I'm Mr. Baker, and this is your presentation for 6.1, DNA is the genetic material. Back in the day, scientists knew that genetic information was carried on chromosomes, and they knew that chromosomes were made of DNA and proteins, but they didn't know which was the genetic material. Was it the DNA? Was it the protein? And trying to figure this out was a race through the 40s and 50s, and so we go back in history to 1928 to Griffith, who was trying to figure out how pneumonia made people sick. It's caused by a bacteria called Streptococcus pneumoniae. The top left picture is a zoomed in electron microscope picture, and the picture on the right is one you would see under a light microscope. He was working with two different strains. A smooth strain caused disease, and the rough strain did not cause the disease. And he infected mice with each strain and recorded those results. Now, how he got smooth and rough is how they looked on a petri dish. There's a video that will explain it much better than I can. But he took the rough strain. Non-virulent is that word. Virulent means causing sickness. Non-virulent doesn't cause sickness. Injected it into mice, and the mice were very happy and lived. He took the virulent smooth strain, put that into mice, and it killed the mouse. So then he cooked and killed the smooth strain, put those into the mouse lives. So now it's the living smooth strain. It's not anything they carry. The bacteria have to be alive. So then he mixed the dead bad guys and the living non-virulent, we'll call them good guys for this, and injected in the mice, and the mouse died. And what's even more surprising is that when they looked in the lungs of the mouse, they found smooth. So the rough strain changed to the smooth strain. And what happened is what we just said. What does it all mean? Well, the rough bacteria was somehow transformed, was the word he used, which is a fancy way of saying changed, into the smooth bacteria. So now, scientists are on a mission to figure out how this happened. But shh, I'm going to spoil it for you. Because it turns out that the smooth screen has something called a capsule. Glycocalyx is a more technical term, but capsule will work just for us. And it's like a protective smoke screen. And they also carry something called a plasmid, which is a small circle of DNA that carry the genes or instructions to build the smoke screen. And if this plasmid somehow gets into the R strain now has the genes, the instructions to build the smoke screen, and they transform or change into the deadly strain. That's what was happening with Griffith. There's a video that we are not going to watch. That takes us to Avery in 1944. Avery then worked on what Griffith did, and he's trying to figure out what was causing it. So he said, well, is it the protein? And so he put proteins in with the R strain and got nothing. Was it the lipids? Put those in with the R strain and got nothing. So then he put the DNA in and he got transformation. And this was another clue that DNA is the genetic material. Hershey and Chase was one of the biggest ones to really seal the deal here. And they worked with little things called bacteriophages, which literally translates to bacteria eaters. These are viruses that only infect bacteria, and they only have DNA and protein. They have a DNA inside and a protein coat on the outside. And they look like the Apollo lunar landers. Here's a good video that you will watch some other time. Here's the life cycle of the bacteriophage. It lands on the bacteria, it injects its DNA. Its DNA takes over the host cell. The host cell produces new bacteriophages. They assemble and then they break out, killing the bacteria. So this experiment was designed to figure out if it was DNA or protein. And so what they did is they labeled DNA and they labeled the protein. And the best way to do that in 1952 was with radioactive material. And so they used phosphorus P32 because phosphorus is only found in DNA. And they used sulfur 35, because sulfur is only found in protein. And they infected the bacteria with these labeled things. And here's a picture. And the red signifies which was labeled. And they did the protein coat. And they figured out that the protein stayed on the outside of the bacteria. So then they labeled the DNA, and they figured the DNA went into the bacteria, which was another piece that DNA was the genetic material. And here's a nice little video on it that you will watch some other time. So what is the structure of DNA? Well, DNA is made out of monomers that are called nucleotides. You should remember this from earlier in the year. They have a sugar, a phosphate, and a base. The sugar is a 5-carbon sugar, and its technical name is called deoxyribose, hence D for the DNA. And the nitrogen base comes in 4 different flavors: adenine, thymine, cytosine, and guanine, or A, T, C, and G. Yeah, adenine, not adenine. That's how they name it in biochem. And so here's your nucleotide structure where you've got the sticky sugar, the phosphate, and the base off to the side. There's your sticky sugar, your phosphate off on the side, and then your base to the inside. And everybody wears sunglasses because we're cool. This is known as a sugar phosphate backbone. It's a spiral staircase, kids. And a spiral staircase has got handrails on the side. That's the sugar phosphate backbone, and it gives all the strength to the DNA molecule. The steps are the bases, and they are not as strong. They come in 2 major groups, the purines and the pyrimidines. The purines have a double ring, adenine and guanine, and double ring, double letter N is how I memorize it, because I always had trouble doing that. Pyrimidines were easy because pyrimidine has a Y in it. So does cytosine and thymine. So that's hopefully will help you memorize it. Then we have to add in Chargaff, excuse me, in the late 1940s. You do not have questions about this in class. That table on the right-hand side, you do not have to memorize. It's just there. And he analyzed the amount of the bases in different species, and he concluded that the number of A's equals the number of T's, and the number of C's equals the number of G's. And this is what we know because A binds to T and C binds to G. And if you have an A, you should have a T. And that little bit down there is optional if you want to write it. Rosalind Franklin in 1951 was doing X-ray crystallography of DNA. And the technique is way beyond the scope of this course, but there is a famous photo, photo 51, and that's how she figured out that DNA was actually a double helix. And she provided that piece, that essential clue that was able to be pieced together to figure out the secret of life. And it's a great example of Matilda effect, where she did the heavy lifting, and of course, men get the accolades. There's a video that you're going to watch, and there's another video that you're going to watch. And Rosalind Franklin takes us to Watson and Crick in 1953, who published the paper where they took all of this data and put it together, and they figured out that you've got the double helix and that you've got A and T bases on the inside. So you have a sugar phosphate backbone on the outside. Here's a picture. Sugar phosphate backbone on the outside and the bases on the inside. DNA is what's called anti-parallel orientation, which means one side is right side up and the other side is upside down. If they were parallel, it would be right side up, but it's anti-parallel. One strand runs from 5' to 3', the other one and the other strand runs from 3' to 5' as you look at it. Now that 5' and 3' is just how biochemists number the carbons in the sugar. You just need to memorize that if one is 5 to 3, the other one is 3 to 5, and vice versa. They are also complementary. It means given one, you can determine the other because the bases always match up A to T or C to G. And here we go. If you've got A on the top, we're going to go across, we're going to add them in. So A to T, T to A, T to A, C to G, C to G, G to C, G to C, C to G, T to A to T, A to T, C to G. I think I may have screwed one up in there, but that is correct. DNA makes up the chromosomes, and this is crazy. This is optional, but you have about 6 foot of DNA in every cell. You have a bunch of cells, and so it means that if I took all the DNA out of your body and stretched it end to end, we could wrap it around the equator of the Earth 2.5 million times. How do you pack that much DNA in a microscopic cell? Well, you got to wrap it around things. Oops. So you're going to coil it around something called a histone to form a structure called a nucleosome. The nucleosome are going to group together to form chromatin. Chromatin supercoils to make what we can see under a microscope called a chromosome. You need to know that histones are the DNA spools. The nucleosome is a group of DNA, and the spools it's wrapped around. Chromatin is, we're going to call it uncoiled DNA, and chromosomes are coiled up DNA. There's a picture to help you conceptualize that. Optional information, a video that you will watch in class, and that takes us to our quiz 6.1. You will need to know all of these things. So study hard, and we will see you next time.